All right, so let's get into it for chapter 10. This chapter is all about biotechnology. I had one student ask about polymerase chain reaction, wanted to know a little bit more about that. So the goal of PCR, or polymerase chain reaction, is to amplify the DNA of a given DNA segment. So we're looking for a certain gene. Okay, so when we extract DNA, right, which is the first step that we're going to do when we're doing pretty much any biotechnology kind of thing, in most cases, we need to get the whole DNA. Okay, and so there's going to be all this entire genome, right, all the genes that you need to make a whole organism. Okay, and you can see that that is going to be a whole big lot of mess, particularly if you're only interested in a small segment of that DNA. Okay, so we need a way to get rid of all this stuff that we don't want so that we can focus on just this gene or a couple of genes that we care about. Okay, so when we do a PCR, right, we're going to put our DNA sample in a little tube, and we have all this other DNA. So let me erase this and draw it in a different way. So here's our DNA in a double helix. And there's all these bases, right? All these base pairs, all the A's, C's, G's, and T's that are forming their hydrogen bonds between each other. Okay. And we don't need all of these. We just maybe need from like here to like, I don't know, here. Okay. So all this stuff here and here is extra and we want to get rid of it. Okay. And so this is going to go on for a long time. There's more and more DNA going off this way that's going off the screen that you can't see. There's more DNA going off this way, but we only care about this one small segment. Okay. And we want to make lots of copies of it because we don't want this to get lost in the noise. So what we're going to do is we're going to do this PCR process. And the first part of PCR, well, let me not say that yet. So when we do our first cycle of PCR, it's going to amplify the part of the DNA that we're interested in and not amplify all the other part of the DNA that we're not interested in. So that stuff goes away, and our target gene goes from 1 copy to 2 copies. And then we do the second cycle. And then we go from 2 copies to 4, and then 4 to 8, and 8 to 16, and 16 to 32. And every time we do a cycle, we're doubling the amount of DNA that we have of just the gene of interest. Okay, so it's a great way for us to be able to, after 30 cycles, right, when we just had 1 copy at the beginning, after 30 cycles, we're going to have 1 billion copies. So having that higher concentration of DNA of that one gene that we're interested in is going to be really helpful for figuring out, like, answering whatever the research question that we want to answer. Okay, so for Terry Mullis, who's the dude that developed this process, he was interested in helping understand what causes sickle cell anemia. And because he developed this way that we could amplify the DNA, now we know that there's just one mutation, right? Another student wanted to know why do we need different temperatures at each of the steps of the PCR process? And it's really comes down to hydrogen bonding. Okay. That first step of denaturing, the reason that we need it to be so hot is to get that, to get all of those hydrogen bonds that are holding the double helix together. We need to break all those hydrogen bonds to allow us to have two single strands. Okay. And so we're going to do that by increasing the temperature, which increases the kinetic energy, which is going to allow those hydrogen bonds to break and give us from one double strand into two single strands. Okay. Once that DNA is opened up into our two single strands, then we can go to the annealing step where we're going to add our primers. Now we remember that primers, so here's our gene. Let's say, oops, A, N, T, C, G, C, A. And then there's other stuff, okay? So our DNA polymerase cannot attach to a single strand of DNA. We need to give it a complement, okay? So our primers are these short sequences of our DNA that are going to allow, that are the complement of whatever our target is. And that's going to create those hydrogen bonds between those two bases. Okay, we want those hydrogen bonds to be formed. So we need to go from being really hot where we need to break those hydrogen bonds to cooling it down so that those hydrogen bonds can be formed. So I'll put here, when we're heating it up, we're breaking the hydrogen bonds. When we are annealing, we need hydrogen bonds to be formed. So we need to cool it down a lot. We are only interested in the genes that are from here to here. Okay, so we're gonna build one primer. That's enough. That's going to allow polymerase to copy this direction. Okay, and then we only need to know from here. So let's go T A. Sorry, A T A. Okay, and that's going to allow polymerase to copy in that direction. So all of this is not going to get copied, right? Anything on the other side of that primer. Anything on the other side of this primer is not going to get copied. And so that's how we can isolate that gene, by having those primers be flanking the gene that we want. That means that when we go to the extension step of one cycle of PCR, or the elongation stage, then we're only copying that part of that gene that we actually care about. Okay. Building the DNA polymer is an anabolic reaction that requires energy input. We're going to use the enzyme polymerase, right, to lower that activation energy, but we're going to also provide energy in the form of heat. It's going to make it easier to actually incorporate those new nucleotides as we're going along and building that new DNA strand. So that's why we kick it back up to about 72 degrees Celsius, depending on your reaction. For your annealing phase, there's a big temperature range because the amount of heat that is required depends on how many A's and T's you have versus C's and G's. C's and G's create 3 hydrogen bonds, therefore they require higher temperatures. So that was something when I was in grad school and we were designing primers, that was something that we had to keep in mind, is that our forward primers and our reverse primers had to fall within the same temperature range, so that we could get both strands of the DNA copied at the same time. Designing primers was not my friend because I'm dyslexia girl. Okay, so hopefully that helps you. So again, I think we talked about this pretty well, I think, in the lecture videos, right? So we do the denature, we do the anneal, we do the extension, and then we come back and we do denature again. And in order for our polymerase to not break down and denature itself at those hot temperatures, that's why we use the Taq polymerase, the DNA polymerase that comes from organisms that are used to living in hot temperatures. In our gene cloning or our molecular cloning, right, our goal is to make a bunch of copies of the gene that we care about or the gene product, right? So, and by gene product, usually what we're talking about is a protein. Okay, so I gave the example of my kid needing the protein insulin because her body can't make it anymore on her own, so we have to get it from an external source. Before we had this process of gene cloning or molecular cloning, in order to get insulin, you had to get after a pig had been butchered, you would pull out the pancreas, put the pig pancreas in a blender, mush it up, and then isolate the insulin from that slurry that you just made, which just sounds nasty. And you can imagine that it takes a lot more resources to grow a whole entire pig than it does to grow a few billion E. coli. So this process has made insulin much cheaper than it was. So 100 years ago, insulin, or like a type 1, a diabetes diagnosis was a death sentence. So, but now we can use this technology to help treat these kinds of illnesses. All right, so let's go through the process. And at any point when you're confused or have needed further clarification, just stop me, okay? Just feel free to interrupt me. All right, so we're going to have 2 test tubes. Okay, we're going to start with 2 test tubes. In test tube 1, we are going to have our plasmid. Okay, and our plasmid is a small circular DNA molecule that just floats around in the cytoplasm of a prokaryote, like a bacteria or an archaea, that's separate from its chromosome in that nucleoid region. Okay, so this is just an extra small molecule of DNA. Usually when we're doing this process, we can synthesize DNA to have this sequence that we like, and to include all the genes that we're interested in. So when we do this in my in-person class, we purchase a plasmid from a supply company and they ship it to us and we can use it. So in our test tube 1 that has our plasmid, what we need to do is add restriction enzymes. Okay, and we saw these before too in our RFLPs. Enzymes, okay. And what restriction enzymes do is they cut the sugar, and I'm just going to write P for phosphate because I always spell phosphate wrong anyway, backbone at a given DNA sequence. So they're going to latch on to that DNA molecule and they're reading and they're like A, T, G, C, T, A. Oh, boom, that's where I'm going to cut. And each restriction enzyme is going to cut at a different sequence. Okay. All right, so that's where we're going to leave test tube 1 for now. let's step over here to test tube 2. And this has our foreign DNA. Okay. And this is our gene of interest. So in the example from earlier, right, this would be the insulin gene, right? That holds the secret code for building a insulin protein. Okay. We are going to cut the foreign DNA, all right, our gene of interest, using the same restriction enzymes. And that's going to leave these little sticky ends, right? So we have this little single-stranded bit that's sticky and out a little bit. Let me turn that so that I don't get lost over here. So this little bit is sticky and out, and that's important because that's going to be the complement. So if this is an A, C, G, this is going to be a T, G, C. And that's going to allow our gene of interest to be inserted into this little hole right here that we made in our plasmid. Okay, so we shove that gene right in here when we put the contents of this test tube 1 and test tube 2 together into the same tube to create this thing called recombinant DNA. So this DNA is a hybrid of both the plasmid and that gene of interest. And does not exist in nature, right? This combination of genes only exists in the test tube that we're working with. All right, so we're almost done with the process, but there's a couple things that we need to do next. Okay, so once we have this recombinant DNA, it's not going to make copies of itself on its own. We need to put it into a living organism. And we're going to put it into a single-celled organism because it's a lot easier to grow those things. And we need all the cells in that organism's body to be expressing those genes. So let's use an organism that has one cell in its body. All right. So in order to have this new plasmid, right, our recombinant DNA plasmid, be picked up, brought in from the environment into the cells of our bacteria, we need to make them competent. And I'm really bad at spelling, and my whole job is to spell live in front of people without spell checks. So good career choice for me? I don't know. Okay, so we need to make these cells competent. What that means is that their plasma membrane, see, is going to become rigid. So that, so this plasmid can whoop, slip in when that cell plasma membrane just opens up just a little bit. Okay, we open up, slide the plasmid in, close it up the cell membrane. Okay, and that at that stage, once those bacteria have picked up that plasmid, that's when they are transformed. They're transformed. Okay, so now these bacteria have been genetically modified and they have a gene, right? These E. coli have a gene to produce human insulin. That doesn't happen in nature. That happens when we make it happen. Okay. And so as this E. coli bacterium is going to be reproducing, it's going to make more copies of this plasmid, so then we can harvest, right, the insulin or whatever the protein is, or right, whatever the product is that we're trying to make. Okay, so after those bacteria cells have been transformed, we're going to grow them on a petri dish that has all the nutrients that bacteria need to grow, but also has antibiotics, which seems a little weird because we're trying to grow bacteria, but antibiotics kill bacteria, so that's weird. But it's not weird if you know that the plasmid that we provided to the bacterium includes an antibiotic resistance gene. Okay, so what this is going to mean, the takeaway message of this is that the bacteria cells that picked up this plasmid that includes, right, whatever our gene of interest is, we gave it a superpower to withstand the harsh environment that we're sticking it into. And the bacteria that did not pick up this plasmid, they don't have the gene that we're interested in. They also don't have this abiotic resistance or antibiotic resistance gene. So when we grow them on an antibiotic plate, they're going to die. So this, by growing these bacteria on this antibiotic media, that ensures that the bacteria that survive are the ones that have all the genes that we care about. Okay? We give them something that we care about, and we give them something that they care about. Okay, and that's how we're going to select for the individual bacteria that have the gene that we're interested in producing the protein for. Okay. So then in the case of the insulin, we grow these bacteria. We don't grow them on petri plates. Actually, we grow them in like these big jars, like big flasks, like kind of like industrial level, right? They would be huge. And from that slurry, right, then you can isolate and purify that insulin and then package it up, send it out to the pharmacy, and I go pick it up so that my kid can stab themselves multiple times a day so they don't die. To help clarify a little bit more, we'll look at an example of what we do with my in-person class. And unfortunately, we just put the bacteria on the plates today, so they haven't grown yet. But if it turns out nicely, I'll take some photos and put it on our Canvas shell too. All right, so what the in-person students did today was we were looking at the gene that makes these jellyfish glow that fluorescent green color, and we gave it to our bacteria. So on that plasmid that we're looking at, so it's called the pGLO plasmid. This gene, this green fluorescent protein, is what causes that glow in the dark. We always have to have an antibiotic resistance gene so that we can select for the bacteria that picked it up and the ones that didn't. But this is a really cool example because not only are we showing this transformation, this bioengineering technique, we also have this really cool gene expression control. So there's this other protein that's coded for on this plasmid called ARAC. We don't have to memorize the names of any of these. This is just an example to give you guys the context so that you can kind of understand this process a little bit more. Okay, but what this protein does is that it will block RNA polymerase when like a certain sugar is absent. Okay. So this is the plasmid, right? This is the DNA molecule that we are hoping that our bacteria are going to pick up. Okay. All right. I think that's all I want to talk about on that slide. Okay. Change my slide. All right. So this is I pulled this picture just off the interwebs. You can Google pGLO results, and this is one of those results. So the students put, they have 4 Petri plates. They have 2 treatments where they put bacteria that were not given the plasmid. They grow 1 on just regular nutrients. Okay. Plasmid ID. Okay. And you can see that there's a lot of bacterial growth on this plate. It looks pretty slimy, pretty snotty. There's a lot going on there. Okay. So that bacteria is just is fine to just grow on regular growth media. Okay, but on this plate, this was no plasmid with antibiotics. Okay. And on this plate, because the bacteria were not given that antibiotic resistance gene, those bacteria all died. Okay, so they weren't able to survive on that. In this case, these bacteria have the plasmid. They are growing on the nutrients and the antibiotic. Okay, but but there's there's none of that special sugar. So we can see that these bacteria are able to grow. But they're not producing this glow-in-the-dark protein. And that's because we have this, without that sugar, this protein, that ARAC sugar, is preventing our enzyme, RNA polymerase, from getting to and binding with this promoter region so that it can transcribe, right? Transcription is blocked in this case. So if we can't transcribe the gene, we cannot translate the gene to produce the protein. Okay. So let's look at this last plate over here then. These bacteria were given the plasmid. They were given antibiotics and all the nutrients. And the sugar called arabinose. And what happens when arabinose is present is it's going to bind with these sites on this ARAC protein and alter the shape of this regulatory protein and get it out of the way so then RNA polymerase can come in and bind with a promoter region, and we are able to transcribe the gene and translate it. Okay, so we're able to see that these are the only bacteria of our 4 treatments that were actually able to produce the protein, because they had to be given the plasmid, the nutrients, the antibiotic, and that special sugar that's going to allow RNA polymerase to get in there and read this gene, this glow in the dark protein gene. And so it's a pretty cool system. It's a little complicated, but I like it because it shows many of the concepts that we've been talking about in this unit. So, all right, so the last question that I had was basically, how do all these processes that we've been talking about in this chapter work together? And then the student also wanted to know what errors can arise, which is interesting because my in-person classes have had a lot of errors this week. All right, so as we kind of mentioned previously, that the plasmid and the gene of interest that we're using in these molecular cloning, we're going to make a lot of those plasmids, that gene of interest, using the process of PCR. Always, whenever you do a PCR, you want to check the quality and quantity of your DNA, make sure that you're getting what you expected. So for example, this plasmid, we know how long it's going to be. We know the number of base pairs that we should be getting. So what we're going to do is we're going to run a gel electrophoresis to make sure that our bands are coming out at about the same size as we would expect. And we're also going to be checking that brightness level, right, to make sure that we do have the proper concentration. So the restriction enzymes, we already talked about those. They're used to form that recombinant DNA. We can use those in that paternity test or that forensics, right? Of those RFLP applications. So there's all of these things, all of these processes can be used depending on what the researcher is trying to do, right? Or what the problem is that's trying to be solved. So they all exist and they all can complement each other. How do you, what's the order of the processes depends on the specifics of the problem. All right, so my last little bit here is what errors can arise? Well, man, my whole grad school experience was experiencing errors in biotechnology. So the one that I pulled up, I was looking for fungi inside of healthy looking plant tissue. And so I had to extract fungal DNA from the plants. So I take the leaves or the roots or the bark or whatever. I think this was bark. And the plant that I was working with are cottonwoods, and they have a lot of defensive chemicals because they have a lot of bugs that eat them. This DNA extraction that I did here, this is supposed to be white, and the liquid is supposed to be clear. So I had a really hard time getting good PCR reactions because I had a hard time purifying my DNA. And every time that you clean your DNA, you also lose a little bit. And I didn't have a lot to start with. So I was kind of like in this catch-22 of like, do I proceed with dirty DNA or do I clean it and perhaps lose the little bit that I have? So some of my samples I couldn't end up using because I just couldn't get clean extractions. So So here's the gel electrophoresis that my students, oh, whatever day it was, um, earlier this week, they ran. And I don't know if you can see the bands very well. Let me go try turning this light off real quick. We'll see if that helps a little bit, if those bands are showing up a little bit better. Okay, this is what this was supposed to look like. So this was our crime scene. Suspect 1, suspect 2, kind of this is what we were supposed to get. And this is what we got. So ignore this is a glare. The only thing that we got was this one DNA band that was right next to the well. So it was like really close to the starting line, which means that it was a really big DNA molecule. And that told me that the restriction enzymes, right, and their job is to cut up the DNA, didn't have enough time to cut the DNA. So these were the same samples that were ran. Well, I put them in the fridge. So they sat in the enzymes for about an hour after this one was ran. And then I had to put them in the fridge so I can go home. But so this enzyme was the same samples that just had more time, and we got the results that we expected. So there are a lot of things that can go wrong. So for my Bio 181 class, we also did the pGLO thing where we tried to get those glow in the dark bacteria. Let me just go back on here for a sec. Yeah, we tried to do this, but it totally didn't work. We got no results. All of our plates looked like this, and none of them glowed in the dark. And it was because we were given the wrong Petri plates. So it's like there's a lot of things that can go wrong. So it's really important when you're working in these with this stuff that you're making sure that you are following proper protocol. You're following proper safety regulations. Like you don't want this E. coli getting on or in your body, getting you sick. Another example of when I was in grad school, my partner at the time worked in the lab with me, and he was, I don't know what he was doing, but he accidentally sequenced his mitochondrial DNA as a contaminant that just some of his cells just flaked off of his skin and landed in his samples. And he got, he was looking for fungi, and he got all this, after he sequenced the DNA, he got all this mitochondria results. And so he found out that he has Neanderthal DNA. So sometime back in his lineage, one of his great grandmas was a Neanderthal. So that was interesting, but it wasn't what the question that he was trying to answer. So similarly, like when I was looking for those fungi in the plants, I got all my DNA sequences back, and some of them were like broccoli and carrots and spinach. And I was like, well, I guess I had a salad that day. So that's one of the things that we talk about when we're talking about that forensic stuff, is that making sure that your protocol and that you're not contaminating the samples with your DNA. Make sure that you're not when you're using your pipette tips and you're transferring samples from here to there, that you're not mixing samples and contaminating them with the DNA of other samples. So there's a lot that can go wrong. But if you're careful and follow the instructions, it usually works out pretty well.